I want to welcome you back to the University of Chicago and thank you for coming this afternoon. I want to take you on a bit of a journey, talk to you about a question that I've been looking at for over 3 decades, and it's really, what is our human nature? And to kind of address such a question, it's useful sometimes to step back and look at nature more generally. NSF has started this Project of Life study where they're trying to document various species. Right now, we've identified about 2 million different species on Earth, but estimates are that there's anywhere from 5 million to 100 million total species, and there's this effort to try to catalog more and more of them. If you look at those species, there's 2, at least in most animals, there's 2 adaptive mechanisms that you find. One, represented by the rattlesnake, is that they're born ready to go. They're able to defend themselves, they're able to fend off, avoid predators, they're able to find prey and eat. The other adaptive strategy that you see quite often from sea turtles to bacteria is that the reproduction is in such large numbers that some of the animals survive long enough to reproduce. And of course, those that reproduce produce genes that increase the likelihood those behaviors would be represented again. Now, a subset of all of these animals are social species. Social species are so defined because these animals create emergent organizations that exist above and beyond the individual animals. That's what we mean by social species. Here you see a sardine, what's sometimes called bait ball or fish ball. Sardines, as you know, swim in schools. When a predator, like a shark or swordfish, comes by, they get all excited. They create these dynamic-looking fish balls. Now, the rule to explain what looks to be this superorganismal process is swim to the middle. You can take your computer and put a lot of dots on it and program them to swim to the middle, and you'll be able to simulate the activity you see in a fish ball. It's a very selfish individual behavior that creates a superorganismal structure. All right? Now, it's selfish in the sense that if you swim to the middle, you're less likely to be predated. It's dangerous to live on the social periphery or to be caught on the social periphery. Those animals, those sardines that swim to the middle, are less likely to be predated, more likely to survive, more likely to reproduce. Those genes that promoted the movement to the middle are therefore inclined in subsequent generations. Now, Darwin understood that there was some process that inclined organisms to have an advantage in reproduction. He didn't know, of course, about genes. And Richard Dawkins, in his 1975 book, The Selfish Gene, kind of captured this with the statement, whatever contributions made by the gene or set of genes to an organism's structure and function are passed on to the next generation, if and only if the gene makes its way into the gene pool. Basically, what this gave us was biological evidence for why there's survival of the fittest in Darwin's work. Interestingly, Darwin knew that there was something else going on. He observed that in some cases, individual organisms put themselves at risk, at disadvantage for survival. And he was curious about why that might be the case. He talked about mutual aid. There was, in fact, a very popular notion of group selection in the first half of the 20th century based on these speculations by Darwin. A graduate student at the University of Chicago in the 50s attacked this notion of group selection, and it fell out of favor, and evolutionary biology really focused on selfish interests from that point on. But over the last several years, we've seen a rebirth, a more refined understanding of group selection processes. It's sometimes called multi-level selection theory, represented here. But for our purposes today, I want to just focus on this particular feature of it, and it is that for species with long periods of abject dependency, and yes, humans have the longest period of abject dependency, reproductive success of an organism is not sufficient. Now, all of the animals I've talked about to date, that is sufficient. It's your ability to reproduce. Those sardines, those snakes, those turtles, when they reproduce, they're more likely to have those genes make it into the gene pool. When you have an organism with a long period of abject dependency, your ability to reproduce doesn't determine what makes it into the gene pool. For if they do not live long enough such that they reproduce, those behaviors are not inclined. And so you still have selfish genes, but those selfish genes have to hijack your brain to produce a different kind of social organism. Now let me give you an example. How many saw March of the Emperor Penguins? Good. So this is review for you. All right? As you know from watching that, emperor penguins are kind of a unique species. They live by the ocean, but when it comes time for mating, they march about 70 miles inland to an even less habitable place in the Antarctica. They mate. They have, in fact, pair bonds that are lifelong. They mate. The mother hatches a chick. The baby penguin has to stay really on top of the feet because it would perish. Both the egg and then the chick would perish if it were on the ice for any period of time. The mother is depleted after the birth, so she marches back to the sea to feed, and the father is left to care for the chick. All right? Folds it under the abdominal fat and basically sits and waits. But you know from watching the show that that isn't sufficient. If that father were to simply sit in the middle of the field where the temperatures drop to minus 70 degrees Fahrenheit and the winds blow as high as 120 miles per hour, if that father were to sit with the chick under its abdominal fat, the father would perish and the chick would perish. So they create a superorganismal structure called the huddle, which you see here. And unlike the sardines, where they're all trying to get to the middle, here they apparently take turns rotating off the periphery of this huddle. Now, most of the time is spent asleep. It's very metabolically draining to sit in such cold weather, but these animals rotate in and out of the middle in a relatively cooperative activity. Right? It is the huddle that is the vehicle for genetic transmission, not the mother and not the father alone. All right? Now, in humans, we also have social structures that contribute to our tendency to survive, reproduce, and care for our offspring long enough so that they too reproduce, but there are slightly different features over the huddle and other animal social structures. First of all, both ontogenetically and phylogenetically, we do not do well alone, certainly ontogenetically. Your offspring would not survive if you didn't care and nurture them, if you didn't protect them. In phylogenetic time, the same can be said. Standing there with a stick fending off great beasts is not a particularly high likelihood event if you wish to survive. We're also, though, incredibly symbolic, abstract reasoning creatures. So our social structures tend to be more flexible, not as rigid as, say, in the huddle or in the sardine ball. They tend to extend beyond time and space. Sounds kind of far-fetched, but if you think about it, you may have children, you may have spouses. If they're not with you, they remain with you in a sense. Their influence remains palpable. If you go to Japan, you find that ancestors play a very big role in their everyday reasoning and behavior and decision-making. They talk to ancestors, former mothers and fathers who might have perished. They remain a part of their life. So they actually have these conversations influencing decisions that they continue to make. They extend to ideological as well as genetic similarity. In other animals, we see kin relationships influencing social behavior. In the Army, you see brothers in arms. You see individuals create these teams and being willing to really sacrifice oneself for the benefit of the group. It's something, in fact, the Army has developed as a training program because it makes the whole group more likely to survive if people have that kind of attitude toward the group. And finally, as you might expect from the way in which I've emphasized their symbolic, abstract reasoning capacities, objective isolation is important, but perceived isolation, whether you feel isolated, is even more important. Well, what's the effect of social isolation? In 1988, there was a science article published on this topic by House, Landis, and Emerson. This is a graph from that article, and what it shows is this is isolated, isolated, integrated on the right. So frequent contact with friends and family, less frequent contact with friends and family. And what you say and see in every study is that the more isolated the individuals, the higher the likelihood of broad-based morbidity and mortality. The explanation for this effect, provided in 1988, was called the social control hypothesis. We know that poor health behaviors increases broad-based morbidity and mortality. I say broad-based because it wasn't just cardiovascular disease, it wasn't just cancer, it wasn't just diabetic complications, it wasn't just strokes, it wasn't just suicides or accidents, it was all of them that were found to be affected by isolation. And so the explanation given by Hauslanditz and Umberson was, if you have friends and family, if you have a spouse, they're more likely to influence you, to exercise good health behaviors. My wife might say to me, John, you're getting fat. Go get some exercise. Quit drinking so much. Get some sleep. Right? Things that are good for me that I might not do if I didn't have a caring spouse. Well, that was an interesting hypothesis, but we suspected it was not right in total, because if you look at other social animals, every social animal I have examined dies earlier when those individuals are isolated. Fruit flies live shorter lives when isolated. Infarct size is exacerbated, and edema is exacerbated in a mouse model where you experimentally induce a stroke if that mouse is isolated. You look at rats who run, you put them on an exercise regimen, they show neurogenesis. If those rats live alone, they don't show the same neurogenic benefits. If you look at rats' response to stressors, those who are housed in an isolated setting show a stronger sympathetic adrenomedullary response. Squirrel monkeys show larger rises in morning cortisol, a very strong stress response we all show as well, if those monkeys are raised in isolation. I could go on, but this suggests it's something beyond a spouse and friends. It's something beyond the spouse telling you to take care of yourself. There's something more direct about the biology and our connectedness. And so we undertook a study of what exactly was going on. How important was connectedness? What did it mean to be a social species? One of the things we found very quickly, because just like in the epidemiologic, we started out by measuring objective isolation. We learned very, very quickly from that objective isolation, when you take a normal sample of people, and we do population-based research, what that means is we take a random sample of people in the United States, a random sample from metropolitan Chicago. We have, in fact, studies of both sizes going on. When you do that, the objective isolation isn't nearly as important as the perceived isolation. Well, I don't know if you remember your first quarter as a freshman, but most people, when they first go to college, leave friends and family behind for the first time. They go to a college where they now have roommates and large classes, and they're physically around more people than when they were at home the preceding summer, but they feel terribly isolated. Loneliness the first couple of weeks is just typical of freshman life. All right? That's a good illustration. Another is when you're an older adult and you lose a spouse, often friends and family are around you. You have more contact than before the loss of the spouse, but one feels like something fundamental about their social fabric has absolutely been decimated. So we looked at perceived isolation as well as objective isolation, and throughout the talk today, I'm going to be talking about perceived isolation. When we put in objective isolation, we find it to contribute nothing additional. Well, it turned out perceived social isolation had been studied by psychologists, and it had a term, and that term was loneliness. So lo and behold, I found out that I was studying loneliness. Loneliness prior to our entry into this field was characterized as an aversive state with no redeeming features. A little different than depression, low social skills, shyness, introversion. We found all of those notions to be incorrect. And in fact, I'm going to share a model, a different theory of what loneliness is. And again, I'm thinking about loneliness or the social isolation, perceived social isolation over an evolutionary context that's now placed in contemporary society. So let's just walk through what I'm going to argue it is. Basically, it's very much like hunger, thirst, and pain. Genes that promote behavior, which increase the odds of the gene surviving, are perpetuated. The selfish gene notion. Selfish genes can lead to social brains because we're vehicles for these genes, particularly when we have long periods of abject dependency. We need to be induced to take care of others. in order for those genes to be likely to be reproduced. Let's say that you and I are out in conditions of privation in evolutionary time. We might pair up and hunt together because together we're more likely to capture a big game than either of us alone. And if we do, neither one of us could eat the whole thing anyway. So it's no loss and a gain for us to do that. So we still are selfish in that action, all right? But the truth is, most days we're not going to find big game. We're going to barely have enough to feed ourselves and still be fairly hungry. Why share? Why share with offspring? Why share with others? Why go back and provide defense when they're in danger? The sardines wouldn't. The snakes wouldn't. Why would we? Well, the argument is we have this genetic variability. Some do, some don't. Initially, many didn't. But mutations started capturing the brains and shaping them differently. Because if none of us were to take care of those others, we may live to go to other contexts and to reproduce more than those of us who took care of their offspring. But the likelihood that those offspring would survive and therefore the genes would make it into the gene pool are increased by those who have this social motivation. All right? Well, one of the means of inducing us to do that is to have aversive cues. Pain is not a pleasant stimulus. Neither is hunger or thirst. If you do not have pain and you do not have modern medicine, you're unlikely to survive. Now, I'm not advocating being in chronic pain, but it's a good thing that you have a pain system because you might roll into the fire in the middle of the night and never know that that happened and perish during the night. Or if you didn't have hunger as a cue, your blood sugar may drop so low before you realized you were running out of energy that you didn't have the sustenance to actually go out and find food. These are aversive cues because they help us survive. Loneliness is an aversive cue that signals something else fundamental about the social organism, and that is our connection to others is at risk, motivating us to do something about that as well. So what does loneliness look like? We all know what it feels like. It's miserable. It's an emptiness that one might have. What does it look like from a mental representation point of view? Now, what do I mean by that? If I asked each of you to describe a room and to describe, to envision a room and to write down or answer questions about it, each of you might describe a different room. You might describe this room, you might describe your study, you might describe your office, you might describe a room in which you spend a lot of time. They would all have different contents. We could then do analyses of these reports, and what we would find is that rooms have 3 dimensions: width, height, and depth. That would be the mental representation, and then stuff. Well, when we ask questions about social connection or social relationships and we apply the same quantitative procedures, we find there are 3 dimensions meant to this mental representation of connection to others. One is what we call intimate connectedness. Gary Larson kind of captures it here. The doc is talking to these 2 designer individuals. Hey, come on, you two were made for each other, and yet there's this separation. Intimate connectedness is the extent to which another individual affirms positive qualities you'd like to see in yourself. Gives you a sense of self-worth. The predictors of that, if you look at a population base that is everyday people, marital status. Those who are married score higher on intimate connectedness than those who are not. Now, it doesn't take marriage to have intimate connectedness. That's a probabilistic statement. As depicted here, people can sometimes be married and be isolated in that marriage. Linda Wade, a colleague of mine in sociology here, has shown that marital status is health protective. Together we've shown that marital status is health protective until you put in how lonely a person feels. If you're married and lonely, marriage isn't health protective. In fact, it's a little worse than feeling lonely and being unmarried. So it's really the fact that marriage causes you to feel connected to others, and that's what's health protective. A second dimension, relational connectedness. This is what people think of when you say loneliness. It's all face-to-face relationship. Your friends, your family, your office mates. You have 2 people on an island together. Thanks for being my friend, Wayne. Well, think about it. You have people in your offices, in your homes. You may or may not, your neighborhood. They may or may not actually be a friend. You may have a lot of contact. What's the quality of the relationship? The kind of predictors of that, frequency of contact with friends and family, is what we find in our population-based designs. The greater the contact with friends and family, the more your relational connectedness. One of the things we found is that people don't limit themselves to connecting to other people. They connect to pets, they connect to gadgets, they connect to God. We experimentally induce loneliness, randomly assign you to feel lonely or not lonely. Your anthropomorphism of gadgets, celestial bodies, pets, and God all increases if we experimentally make you feel lonely. It's social connection through inferential reproduction. Third, collective connectedness. These are social identities. You have two individuals that happen to be bears. They're sitting on, say, Michigan Avenue here, and they're obviously close to one another and friends, and one looked at the other and said, well, we're lost, and it's probably just a matter of time before someone decides to shoot us. It's a sense in which you don't belong, or you do belong. If you remember junior high and not being in the right clique or on the right team, or being on the right team but being the last one selected for that team. Or think about 9/11. That tragic event was followed by a sense of harmony and cooperation in the United States that was so profound it made media. People were talking about how everyone was kinder to one another. Why was that happening? Well, the reason it was happening was because of this collective identity. That tragic event caused us all to re-identify as having something in common with one another, as being a member of the same group, and that is Americans. And that led to this, if you will, increased altruism and cooperation within the group. The best predictor of this facet is the number of voluntary associations to which people in America belong. So that's, if you will, the mental representation. What is loneliness? Who are the traveling partners of loneliness? These are data from Ohio State University undergraduates, 2,500 to be approximate. And we took the top and bottom quintiles of these undergraduates, and we then looked at what else varied with loneliness. And I put it on a common scale, so all of these differences that I'm showing you are statistically significant, in fact, fairly large. Loneliness does not travel by itself. Lonely individuals tend to also have poor social support. They tend to be shyer, poor social skills, somewhat hostile, anxious, low self-esteem. They fear negative evaluation. They tend to be pessimistic, low positive affect, high negative affect relative to those who feel connected. Well, who's the driver of this bus of characters? Is it loneliness or is it one of these others? Is it the negative affect? Is it the depression? Is it the low self-esteem? What's driving? Well, the way we answer that is one of two means. One, we do experimental studies. We randomly assign people to feel lonely or not lonely through various nefarious manipulations we've come up with, and I'll show you an example in just a moment. And we do that when the question we're asking has a short-term answer that's not dangerous. But in truth, we want to understand in part, how does isolation kill you? We can't, we shouldn't, and we don't randomly assign people to feel lonely over a 10-year period to study to see if they die sooner. For that purpose, we do longitudinal studies. And we're currently in year 8 of a longitudinal study of individuals here in Chicago metropolitan area. Half men, half women, a third Hispanic, a third African American, a third Caucasian. They get tested, in fact, just across the sidewalk in the Institute for Mind and Biology on the top floor. So we bring them in once a year. They walk in with vats of urine. We test them all day, physiologically, medically, psychologically, behaviorally, and then they leave with test tubes from which we get salivary cortisol and other endocrines. And then we measure and we draw blood while they're there so we can do genetic analyses. And we've been doing this. And so we have longitudinal data to look at variations in a variety of states. I'll show you some of them. And that allows us to see what are the negative physiological consequences and psychological consequences of loneliness over time. The experimental study. What we did was to take a group at Stanford. David Spiegel is a head of psychiatry out there. He had written a book called Experimental Hypnosis. He's in fact the preeminent experimental hypnotist in the nation as a researcher. He had just published a Lancet article showing that if you take high hypnotizable subjects and show them cubist art that's in color or black and white, and you tell them orthogonally, You're looking at something in color or black and white. The brain's response reflects what they were told under hypnosis, not what they were looking at. If you look at low hypnotizable, the brain response reflects what they're looking at, not what they were told. Now that impressed me. David's a friend, so we flew out and we used those same subjects, and David did the hypnosis. And we induced in the same individuals feelings of isolation. Feelings of isolation are feelings of connectedness. And I have exactly the same scale, and you see the consequences. It was absolutely stunning. We did not expect the fingerprint to look this similar. Loneliness is the driver. And we did this study over a decade ago, and it was really this study that made me think, wait, this is not just some associate, some variable that's driving along with all these others. This is something much more central than any of us have realized. And it was at that point that we started to get a lot more serious about figuring out what was this variable and what was it doing. So what I'd like to do in the rest of the time is simply go across different levels of organization, starting with demographic and going down to genetic, and showing you what we've learned about how loneliness operates and what's happening to loneliness in our world. Okay? What I have here is a map of the United States in 1940, and the color represents the percent of residences in which a single individual lives. And the light blue indicates that there's fewer than 10% that are living in individual households in 1940. 30 years later, 1970, you see it's a little bit darker, in fact considerably darker. You're in the area of 15 to 20% of the households in most states now have people with single individuals inside. 30 years later, 2000, it's over 25%. Now, there's reasons for this, and some of the reasons are good. We have an aging population, and we're living longer, so the length of time spent in widowhood is longer. Therefore, more of us are sitting there in single households. If you don't live very long, then that turnover is faster. This is not a bad thing, but this is the consequence. We're a more affluent society. 60 years ago, if you lost your spouse, you may well have had to move in with family and friends because you didn't have the resources to live alone. Now we do. There's reduced intergenerational living. There's increased geographical mobility, making it less likely we have friends and family around with whom we could. We have extended life with disabilities. Even when I limit this analysis to 35 to 45-year-olds, however, we find increased solitary living. Well, there's delayed marriages, delayed families, which are contributing to this as well. The fact, however, is that we're living in more isolated environments than we have in the past. What about cultural? Well, this might surprise you. There's what we call collectivist cultures and independent cultures. An easy way to think about what's a collectivist versus independent culture is watch how they eat. In China, when you go to a dinner, go to any restaurant and watch them, you'll see huge lazy Susans, and they share food, right? If you're not attending to other people at the table, just trying to spin to get to what you want, you'll see chopsticks flying across the table as somebody was dipping in and got it jerked away from their hand. You have to pay attention to everyone in order to know how to eat and not interfere with others. In Italy, you pass around big bowls of food and everyone shares, family style. In America, we do drive-throughs and we get our Happy Meal in our little box and eat by ourselves. Right? Collectivist, individualist. Who's lonelier? Well, it turns out loneliness is higher in collectivist cultures. Surprising at first, but remember, most people do not feel lonely. Loneliness is the exception. Most people don't remain lonely. Loneliness is the exception. Just like most people don't remain hungry, thirsty, or in pain. It's a cue that motivates us to do something to remove that cue. Now, loneliness, I would say, in our society has been stigmatized. It's not understood to be like hunger and thirst. It's thought to be some kind of personal weakness. It's not, but that's how it's conceived. If hunger were so conceived, we'd have more anorexics walking around. So we have more individuals walking around in lonely states than we probably need. Why this particular effect? Well, within our individualistic culture, when do you find people feeling the loneliness? was the loneliest? Easy. It's on those evenings when it's the cultural norm to be together with friends and family. Thanksgiving, Hanukkah, Christmas, Easter, those kinds of evenings when the expectation is you're with others. If you're with others but in disagreement with them, or you're not with others, loneliness spikes even in our culture. In collectivist cultures, it's like Thanksgiving every day in terms of the social expectations. So when you find yourself disconnected, the pain is heightened because you compare yourself with what you believe you should have. What about economic? And to be quite frank, I worried about this. I worried that what we would find is the lonely individuals are the funny-looking, less intelligent, heavier, shorter individuals. So Michael Jordan would be the opposite. Handsome, tall, attractive, wealthy. I mean, the man has social capital. He walks in, there's all kinds of, if it's a meat market, there's all kinds of exchanges he has to offer. All right? And so I worried, as you sometimes see depicted in the in the media, that lonely kids, lonely adults are those with less social capital, and it's just an economic exchange process. We found absolutely no evidence for that. None. Loneliness isn't related to income. It's not related to height, to weight, to BMI. It's not related to physical attractiveness. We had dramatic differences in physical attractiveness in the studies, but the attractive individuals were just as likely to be lonely or connected as the unattractive individuals. We did, and others have found an effect between loneliness and economics. And Charles Dickens actually anticipated this in A Christmas Carol. Money makes you more likely to want distance between yourself and others. So in a study that Katie Vose did from Minnesota, she put up a computer and had on the computer a screensaver, and that screensaver were either fish or this, dollar bill signs, floating across the screen. Afterwards, in one of a variety of studies, she measured how close you stood to someone else, how much you wanted contact with others, how helpful you were to others, and how aggressive you were to others. Those who had been exposed to the dollar bill screensaver, which they were not, it's just something that was in the background, but those who were exposed to the dollar bill wanted less contact, stood further apart, were more hostile and less helping of other individuals in different studies that she did. What about social context? Well, this is a study published by McPherson and colleagues a couple of years ago, and what they did was to repeat questions asked in 1985 in the United States, and what they found was that frequency of contact with friends and family has decreased over the past 20 years. Given the demography I showed you, that's probably not a surprise. They asked one other question, and they said, how many confidants do you have? The most frequent response 25 years ago was 3. In 2004, the most frequent response was 0. It's a dramatic change. We looked at the Framingham study. Framingham, Massachusetts. People have been following this group for more than 40 years now. It's given us enormous amounts of information on the etiology of various cardiovascular disorders. Less well known is we also have social network data on those individuals. We know who people's friends are, and we've known for a number of years. And so what Nicholas Christakis, James Fowler, and I did was to look at where is loneliness in this network. And what you find is loneliness lives on the periphery. More interesting is that it doesn't start in the periphery. It starts on the inside, and it becomes contagious. And over time, you lose friends and family and move to the periphery, but you leave behind others who now feel lonelier. So this is the percent of linked participants who felt lonely. That is, you're in this network, you're in the middle. How many of your friends and family felt lonely? How lonely did you feel 2 years later? And what you see is the more people in your context who felt lonely, you are more likely to feel lonely later. It's contagious. Moreover, we found this effect up to 3 degrees of separation. So let's say you and I are friends and I get lonely. Okay? You're more likely to be lonely. Let's say you know this gentleman, but you don't know me. You're more likely to be lonely. You don't know him, but you know her. You're more likely to get lonely because of my loneliness. Now each one, you show the biggest effect, you show an intermediate effect, you show a smaller effect, all of them are above 0. You may know him, you're not at risk. By the 4th degree of separation, it's at non-significantly different than 0. But 3 degrees of separation. So you have this perpetuation of loneliness in these social networks. What about behavior? Well, in young adults, lonely and non-lonely don't differ. When you get older, we start to see differences in the health behaviors. Lonely individuals in particular are more likely to exhibit behavior that's immediately rewarding but bad for them. So for instance, the percentage of calories that's in fats and sugars is higher in lonely than non-lonely older adults. If you look at exercise, well, first of all, if we look at older adults in the Chicago metropolitan area, I was surprised how little they exercise. I was also surprised by the level of obesity that we have in Chicago. But the lonelier you are, the less likely you are to be exercising, and if you quit exercising, the less likely you will start again. Now remember, the health behaviors is something that House, Landis, and Umberson suggested was accounting for broad-based morbidity and mortality, and it existed because of the social control hypothesis, friends and families influence. We tested that. There's another effect of loneliness that we've documented, and that is loneliness makes you dumb. Loneliness impairs executive functioning. Yes? Great question. Older adults, well, I now fall in this category squarely, all right? We're studying individuals born between 1935 and 1952. So when we started, they were 50 to 67, and now they're about, yes. But we see it on a continuum, so if you're 40-ish, you're not, this doesn't save you. It just means it's not as strong. 30-ish, whatever. Okay, so we tested whether it was social control or whether it was this impairment in executive functioning, and we found it was the executive functioning. It wasn't that friends and family were influencing you, it's that when you have this horrible, miserable day of feeling isolated and lonely, and you go home, are you going to eat the cookie or are you going to go out and run? Well, if you had a really bad day, it's really easy to have the cookie and not go out and run. Yes, you had a question. Yeah, what you do is to test that is you flip those two variables. When you do that, you don't get it. We also have longitudinal, which we test these with as well. So it's a great question. When we do it longitudinally, again, we see your executive functioning this year predicts exercise next year. That is, loneliness is mediated by the executive functioning, which also gets away from that problem because of the temporal differences. Good question. Thank you. Please feel free to ask questions. I welcome that. Okay. Well, one of the notions that has existed is that loneliness and depression are basically the same thing. If you look at some common depression measures, items in the scale include, I feel lonely. Well, we and others have demonstrated that loneliness and depression are two different states. They're statistically separable. They're functionally separable. Here's a case where we have 5 years. So L1 through 5 is loneliness in years 1 through 5, depression in years 1 through 5. We have a variety of demographic variables we're going to take out. We have a variety of medical variables and drug variables we're going to take out. And so what I'm going to look at is, can I predict the change in depression above and beyond what's predicted by all of these variables if I look at the prior year of loneliness? It's called cross-lag panel analysis, but I'm looking at the residual variance, what's left in the change in depression above and beyond what depression can predict, and above and beyond with all of these. And I'll tell you, we also added into these your social support, the stresses to which you've been exposed, and your hostility. So we put all of those in, and we find the same thing I depict here, and that is loneliness predicts increases in depressive symptomatology. And that's true year by year. When we add those other psychosocial variables, the ability of depression to predict loneliness disappears. So it seems more unidirectional than we had actually anticipated it would be. Loneliness makes people depressed over time. Have you ever walked through the woods to enjoy the leaves, only to find yourself kind of startled, look down, your heart beating hard, and see a curved stick? Has that ever happened to any of you? Walk through the woods, startled, look down, and there's a curved stick. Basically, you have a man walking through the woods, He actually stumbles upon a snake, but a curved stick will do the same thing. We are evolutionarily shaped to look for dangerous snakes lying in the grass. We don't know we're out walking around looking for snakes. It's something that evolution has sculpted our brain to do, and it does it without our awareness. Well, loneliness, we argued, has a similar effect on what your brain is doing that you're not privy to. And that is, it causes you to look for social threats, but you're unaware of it. It puts your brain on high alert for social threats. Now, how would you test that? I'll show you. What I want you to do, I'm going to show you a set of words. The words themselves are unimportant. I want you, as soon as I show this, to think to yourself and to name the colors in which each word is printed. It's the colors in which the words are printed that I want you to pay attention to, okay? So I think I have 4 or 5 words. Just think as fast as you can, what color is each presented in? Ready? Okay? Straightforward, easy. I'm going to do it again. Same thing, same task. It turns out there's 4 words. There's going to be 4 again. Ready? Harder, right? Okay, all you wanted to do was to look at the color, but your brain didn't allow you. Your brain is reading the words, and that's messing you up. So we use this interference effect to examine what is your brain doing that you don't know it's doing. And so what I'm going to do is I'm going to put, instead of green, blue, orange, red, I'm going to put positive words like gift and cooperate, and negative words like vomit and reject in a list, and we're going to show them to lonely and non-lonely individuals, and they're going to name the colors in which the words are presented. If loneliness puts you on high alert for social threats, lonely individuals should be uniquely slow in naming the colors of words like reject when we've controlled for how long it takes to name the color of words like vomit. All right? Here's what we find. Negative words, lonely individuals are slower than our non-lonely subjects in naming those colors. Positive words, no difference. Their brain is on high alert, but they're unaware of it. Now think about what this means lonely versus non-lonely people see in their world. Have you ever been hungry and you've driven down the street versus non-hungry? When I do it and I'm hungry, you know those golden arches? Usually I ignore them, they jump out. Right? Well, if you're lonely and your brain's on high alert for social threats, similarly, the social environment in which you operate looks different, feels different, is different, affects cognition and behavior differently. One of the things we questioned was, does this brain being on high alert for social threats penetrate the night? You know, we think a lot about the stresses of everyday life, taking a wear and tear on our bodies. Well, we're not only an organism that has wear and tear. We also repair and maintain ourselves. We have a repair crew called anabolic physiology within our confines. Is anabolic physiology compromised when you feel lonely? Why might it? Well, remember I suggested if you had a stick fending off wild beasts, that would be a dangerous circumstance. Well, if that's dangerous, imagine putting the stick down to lay yourself down to sleep without the safe social surround, and you have predators out. Indeed, what we found was lonely individuals show more micro-awakenings and poor sleep quality. Micro-awakenings. You're unaware of these. Just you wake up more exhausted. Your spouse might be aware of them. Sleep apnea patients show this. They're tired in the morning, but they're unaware of the fact that they were snoring or breathing, and then they went, Spouses tell them that they have sleep apnea, because it awakens the spouse sometimes. Now, this isn't quite as extreme as sleep apnea, where they're gasping for air, but it's the same kind of micro-awakening where they arouse and then they fall right back asleep and they're completely oblivious to it. That's what we found in the sleep studies, and we find, in fact, that in reports, these individuals complain about daytime fatigue. Now, many of the studies I'm showing you, we looked both cross-sectionally and we looked longitudinally. One of the things I've learned, and I showed you with the experimental hypnosis study, is that I used to think lonely and non-lonely were different people. What I've learned is we are different people when we feel lonely versus non-lonely. within individuals, we see effects as large as when you look across people. Here's a case in point where it's the same individual, their isolation, their sleep dysfunction, day by day, in a cross-lag panel analysis with the same covariates I showed you before. That is, we're controlling for the same kind of other variables. And what you see is how isolated, how lonely you feel in the evening predicts that night's sleep dysfunction and not the other way around. Now, truthfully, sometimes I have grant deadlines, and I'm up for days working on these grants before the deadline. By the time I've gone several days without sleep, I am not as nice a social creature. But those are relatively rare, particularly when you look at a population-based designs. I would suggest that people sitting in here and people like myself aren't your average person. The average person doesn't show enough sleep fatigue that they actually produce that kind of impact on social behaviors. It's the other way around. Well, remember I said if you have your brain on high alert and you're unaware of it, just like when you're hungry and you're unaware of it, your world looks different. In fact, it does. These are data from High and Low Lonely, and we're looking at what kinds of uplifting events and hassles are they experiencing. And what you see is there's no difference in the lives of lonely and non-lonely young adults. But their interpretation of those events differ. The lonely, and these turn out to be the same events. This was our undergraduate study. We're beeping them 9 times per day for 7 days. And what we find is, even though the event is the same, the lonely individuals complain of the hassles being more severe and the uplifts less uplifting. So we then did a brain imaging study, so we're down to the neural level now, to see what's going on. And what we find, well, let me show you the study first. So we show a lot of pictures that are social or non-social. We subtract. These are equally visually complex, they're equally extreme, they're equally arousing. We subtract the hemodynamic response we see to the social pictures and the equally unpleasant non-social pictures. We then take the level of loneliness and see how that brain differs in a lonely or non-lonely mind. What's the brain doing differently? Okay, what you see back here, these yellow areas, for those who are not used to seeing this is the back of the head, this is the front of the head, this is the right side, radiology kind of standards. You see these 2 yellow patches? That's the visual cortex. These are pictures. It shows that lonely individuals are paying more attention. Their attention is riveted when it's a negative social picture, when it's a person. And remember, they're on alert for social threats. Here's another evidence that you show a social threat, they pay more attention than when they feel non-lonely. If you follow the visual stream forward, you get to an area called the temporal parietal junction, designated in blue on the right and left side. The temporal parietal junction is involved in something of a miracle of the mind. It allows you to control your attention to go from inside your head to take the perspective of someone else's head. That's something of a miracle. Non-lonely individuals are more likely to do that when looking at someone else in distress. Now, after the fact, this actually makes sense. If your brain feels threatened, right? If it's already on high alert for social threat, here we have a dangerous circumstance, you don't have the luxury of taking someone else's perspective. You're in danger. You have to look out for yourself. Of course, if that's your state of mind, you're going to have less understanding of the other person, and you're going to be less likely to pull out the social skills that are most appropriate for that circumstance. So indeed, when we look at social skills in older adults, they have the same social skills, but they don't pull out the appropriate social skills with the same likelihood, with the same probability as someone low in loneliness, who's more likely to take the perspective of the other person and understand what they're feeling. When we did that for the positive, same approach, we found a very different area of brain activation. This is a very large area centered on the ventral striatum. The ventral striatum is a dopamine-rich area of the limbic lobe responsible for reward. When you feel reward, when you see someone you truly love, when you eat a delicious piece of chocolate, when an addict sees the substance to which they're addicted or consumes it, you get activation in this region. It's the non-lonely who are showing greater activation of this region to a person than to an object. Now, lonely and non-lonely show activation of this region to both pictures. They're pleasant. They're felt to be pleasant. But the non-lonely shows stronger activation to people than objects, whereas the lonely don't. Remember I showed you that the uplifts were less uplifting to the lonely? Their brain. They actually have less dopaminergic activation in response to those social uplifts. Okay, what about autonomic? These are young adults. We stress them. In fact, it was a social stressor. It was a speech stress. And what we find is lonely and non-lonely show increases in blood pressure. Well, as many of you may know, blood pressure is a regulated endpoint. You can't afford for this to go out of whack. It's harmful for your health. And so when you're young, those homeostatic mechanisms are intact. It controls your blood pressure. No difference as a function of loneliness. Blood pressure you can think of as the pressure coming out of a garden hose. You can increase the pressure either by turning up the amount of water coming out of the spigot, or you can put your finger over the end and increase the resistance. Both will increase pressure. When we look at how is that pressure being achieved, we find a difference. Lonely individuals show that blood pressure, but it's by higher vascular resistance. Right? But notice that higher vascular resistance is there at baseline. Not only in response to the stressor, it's there at baseline. This is interesting for a couple of reasons. One of which is if I make you feel threatened, I put you in a room, I set up the circumstances where there's a bear outside and you can see it and it's trying to get to you, right? And slowly but surely, it's tearing down the door. You'll show high vascular resistance. Feeling threatened increases vascular resistance. Well, I've suggested this brain is on high alert for threats all the time. That's what this is suggesting. Well, they were in the hospital. These are kids. Maybe they felt that it was the circumstance of examination. So we had developed an ambulatory impedance cardiograph and blood pressure device that fit into basically a camera box. So we put this on individuals. They went about their normal lives. 9 times they were beeped. They sat down. They pressed these buttons, and we were able to do the same measurements we were doing in the hospital during their everyday life. And what we find is blood pressure, and they're lonely or non-lonely. During a normal day, it's the same, but they have higher total peripheral resistance, vascular resistance, and lower cardiac output. Again, this is interesting because blood pressure is complexly determined, but one of the determinants is vasoconstriction, this vascular resistance. The Framingham study, in fact, informed us that if you look at what is the predictor in young adults of late life hypertension, it's vascular resistance. So we then said, do we find higher blood pressure in our lonely older adults? And those are the data. The answer is yes. And that's a cross-sectional. The next question is, if we look longitudinally, do you show elevated blood pressure down the road if you feel lonely when we start our study? Here are the data. Very much like the depression, but we have longer lags because, again, blood pressure is regulated. So we didn't expect the effect immediately. We thought it would take longer for loneliness to, if you will, penetrate all the protective mechanisms that control your blood pressure. And indeed, by year 4 and 5, we find earlier levels of loneliness are contributing to later elevations in blood pressure. This is changes in blood pressure over what you saw in year 1. So they started out with higher blood pressure here, and they're also showing bigger increases over time. This is not a trajectory you want to follow very long. Okay, let's go to the neuroendocrine. Back to this map, you see that one of the antecedents of vascular resistance is cortisol, a very powerful steroidal hormone. Indeed, we find morning rise in cortisol to be higher in our lonely than our non-lonely subjects. Remember the squirrel monkeys showed higher morning rises? So do you. Right? By the way, when we look, this is cross-sectional. When we look longitudinally at you, when you feel lonely or non-lonely, or relatively lonely or relatively non-lonely, we find that predicts, even within subjects, within each of you, the magnitude of the morning rise in cortisol. The other variable that we found related to loneliness, we're very careful about what else might it be due to. I haven't been discussing this, but we control for a host of other factors. In this study, we found daytime fatigue was also related to that rise. What you see longitudinally is it's not daytime fatigue that's leading to higher rises. The higher rise leads to lower daytime fatigue. Yes, carbohydrates and sugars, fats are broken down in part by cortisol release. So if you have a big cortisol pulse, you feel less fatigue that day. But loneliness seems to be what's causing that rise in cortisol. Let me go to the neuroendocrine a little bit more and kind of provide a quick summary. This is essentially the whole system that we've been talking about. Out of the hypothalamus, we have the corticotropin-releasing hormone acting on the anterior pituitary, releasing a corticotropin hormone release. This ACTH is actually higher in the morning for lonely than non-lonely. You might expect that since that's underlying this higher morning rise in cortisol. Cortisol circulates through the bloodstream, acts on the brain in ways that subsequently can compromise cognitive functioning, and loneliness has been found in 3 different studies now to predict cognitive decline. One of the studies I just read yesterday showed your IQ at age 11, predicting your IQ at age 79, 488 people. At age 11, they had measured 87,000 people in Scotland. They were able to track down 488 of these individuals when they were 79. They controlled for age, IQ at 11, social class, education, and they examined a number of variables to see, including social activity and social support and stress, what predicted IQ at 79. The only variable to predict it was loneliness. So this operates back on the brain, but you notice we have the immune system in here. All these leukocytes are being bathed in blood with elevated levels of glucocorticoids, cortisol. These leukocytes have receptors on them for cortisol, and they have nuclei in them, creating transcripts. The DNA is being translated into RNA. Can it be that the social world actually modulates DNA to RNA transcription? We asked that question in collaboration with Steve Cole, and we looked at 3,300 different gene transcripts. 10 years ago, I did a study. We were able to look at 1. Now we're able to look at 3,300. And we found about 200 different. These are the transcripts. About a third of them were overexpressed, and about 2/3 were underexpressed in lonely individuals. Now these, think about them like letters. You don't want to try to interpret 200 different transcripts. Instead, there are bioinformatics tools, we use TELUS, that look at these transcripts as if they're letters and putting them together to see what words or messages are being expressed based on the functional similarities of these transcripts. And we found something that makes perfect sense in light of the graph I just showed you. These leukocytes have glucocorticoid receptors, just like your eyes have receptors. Well, if you're in a very bright context, your eyes diminish their sensitivity to that luminance. If your leukocyte receptors are exposed to high levels of cortisol, as is the case with isolated individuals, the receptor transcripts that take that message and signal the nucleus decrease. So there's a reduction in those GRE transcripts. Now, one of the things cortisol does is to break down fats and carbohydrates to provide energy. Another thing that it does is to control inflammation. This is why pitchers get corticosteroid shots in their elbow when they're pitching. Brings down inflammation. Well, if the nucleus of the cell isn't hearing these cortisol signals, the brakes on the production of pro-inflammatory cytokines is reduced. NF-kappa B transcript family are pro-inflammatory transcripts that then lead to inflammation throughout the body. That's upregulated in our lonely individuals. So you actually get a difference in your leukocyte distribution. Finally, is loneliness heritable? You might expect it to be. And remember, heritability is about individual differences, not high or low levels. It's about individual differences. And I would argue we need variability. We need individuals who go out and explore and discover new territories and are not so bound by the pain of social disconnection that they can't do that. Others who are bothered by that and are more protective, if you will, of the social surround. When we look at it, we looked at this with Dorette Boomsma, who's the director of the Dutch Twin Register. We looked at about 8,400 people. We find that loneliness is about 50% heritable and 50% environmental. What is the genetic substrate? Well, there's a number of different genes. There's not a single one. Oxytocin receptors seem to be involved in this, but I'm going to show you one that actually underscores a point I'm trying to make in this talk, and it's the ApoE4 allele. ApoE4 predicts Alzheimer. If you have this particular allele, you're at risk for Alzheimer's disease. It is related to brain lipid metabolism and, in fact, an impaired brain lipid metabolism. Keely Chow and I did a study, this is Taiwan now, looking at individuals with no unmet social needs or with social needs. We actually operationalized this two different ways. We get the same results. And we're looking at those who have this E4 allele and everybody else in the sample. And what you see is whether or not you have this allele, loneliness is relatively low when you have no unmet social needs. The argument, just like hunger, thirst, and pain, is not that what is heritable is a high level, it's a sensitivity. Some people are very sensitive to being hungry, others less sensitive. Very sensitive to being thirsty, not so sensitive. Very sensitive to a nociceptive, a painful stimulus, or not so sensitive. Here the argument is some people are sensitive to when social needs are not being met versus not so sensitive. No unmet needs, no provocation, low levels of loneliness. What about when there are unmet needs? What you find is those with normal brains show increases in loneliness. This is the normal response. It's those with the E4 allele, the compromised brains, that don't show that increase in loneliness. So I end with this because it shows you that loneliness is in fact normal. It's actually made me think very, very differently about our species. Something like this contributes to, if you will, our humanity, the fact that we're willing to go to personal cost for the benefit of others, in part because it's reinforcing to us. It moves our pain and is in fact reinforcing when we do so. So, there's this regulatory loop that loneliness instantiates. When you become lonely, or when you perceive yourself to be socially isolated, that's a dangerous situation. Ontogenetically and phylogenetically, leads to implicit hypervigilance for social threats that biases your attention, the kind of hypotheses you're testing, what you remember. That leads to behavioral confirmation processes. If I'm looking out and I see social threats everywhere, and we interact and you're just not particularly kind, but you're not nasty either, I might see in it a more sinister intent. If I expect you to be a nasty person, I'm going to act in a way that's protective and paradoxically self-defeating, which then, of course, leads to more negative displays, that is, expressive displays, more negative behaviors, more negative interactions, more negative moods. We've demonstrated that as well. Of course, our social environment is a big part of this. I might have loving partners and family who can get me out of this, or I might have less nurturant social environments, which will simply perpetuate this, and you go round and round. And of course, in the middle, we have neurobiological mechanisms that contribute to increased morbidity and mortality. So how does that morbidity and mortality come about? There's 5 transduction pathways through which it operates that we've talked about. We have these genetic and environmental substrates, and I hate to say, we have this physiological resilience that goes down with time. We're physical devices we wear out. The reason people my age have influenza vaccines is older adults die of influenza. Young individuals, they're very unlikely to die of influenza because their immune system is so efficacious. So the recommendations are those older have flu vaccine, not those younger. This operates across systems. As we age, this decline will occur, but the slope is not invariant. Typically, the hypothalamic pituitary adrenal cortical axis is exaggerated in lonely individuals, or when you and I feel lonely, speeding that decline. And finally, anabolic physiology. Sleep quality, the restorative aspects of our everyday lives, are diminished so that the toxicity of the days are less detoxified over the course of the night, cumulatively leading to a sped decline. And through that process, producing higher rates of morbidity and mortality. And notice it's broad-based morbidity and mortality. So to bring us full circle, I think there's something of a parallel in what I've been talking about and how we used to think of celestial bodies. If you look up, it sure looks like we're the center of the universe, doesn't it? Like it's just us and everything circles the earth. And if you look at our own eyes. Our brain is seated deep within the cranial vault inside each of our heads. And so it's easy for us to think of our minds as individualistic, isolated. It's just about me. What's good for me is good for society. But neither actually operates that way. That may be what it seems like inside your head. But we now know there are invisible forces acting on the earth, gravitational forces, cosmic rays, a number of different processes, and we're part of a much broader, larger system. And I would argue the same is true of our minds and our bodies. They were part of a social species fundamentally, and it is that social species that contributes to who we are as humans. Thank you very much. It takes a village, by the way, to do this kind of research, and these are among the collaborators who have contributed to this. Thank you for your patience. Yes.
I gather that sleep, lower sleep quality is bad. And you compensate for that by more sleep quantity?
Yeah, good question. We look at sleep quantity and quality. There's no difference between lonely and non-lonely. One of the things we find in older adults is they're taking more, the lonely older adults are taking more sleep meds. These are individuals who've had poor sleep for a long period of time. The sleep medication acts only acutely, and so that's not helping. They may sleep longer, but they're still waking up more exhausted. Yes?
One of the social, you reported on studies that show decline in social networks.
Yes, yeah.
I wonder if you think electronic social experience.
Great question. Yeah, so the question is, what about the internet, and what about digital connections? We've actually looked at that, and the answer is straightforward, but it's a little bit. The underlying theory is exactly as I posited. If you're lonely in everyday life, you tend to be lonely online as well. If you use the internet as a replacement for face-to-face, face-to-face is rich in terms of its connections. If you use it as replacement, use of the internet actually makes you feel lonelier. Now there's one exception. If you're isolated because of some disability, or because of a spouse who has Alzheimer's, for instance, so you don't have the opportunity you used to get out, the internet has provided a huge relief for those individuals because now they can connect with others of the same situation and able to share where there was nothing to share before. But the person who takes all their time, garnering 4,000 friends on Facebook, they're probably very, very lonely individuals. That's basically what we find in this research.
Does that depend on what mode they use online?
Pardon me?
Does that depend on what mode you use online? So if it's text-based communication versus Skype-based kind of communication?
No, it actually doesn't. It's across different because it's really, one of the things people think is, to solve loneliness, put them in groups. So, and it turns out that doesn't actually lower loneliness. Why? Because of that loop I showed you. People who go into group therapy as lonely individuals, everybody thinks that it lowers loneliness. We just finished a quantitative review of all the studies that have been done. It doesn't do a thing. When you look at those studies, the effect sizes are not different from zero. And that's because they start out in this group and feel better about it, but then they start interacting in ways that are more hostile, and over time, Skyping with somebody you don't like very much doesn't make you feel less lonely. Yes?
Has a study like this been done on a prison population?
There has been a study, not exactly of this quality. I read one just this weekend on a prison in some other country, can't remember, I think maybe South America somewhere, and they find, here's a shock, the prisoners feel lonelier than the guards. They also feel more threatened and more hostile. So yeah, that's what's been done in the prisons that I've seen. People have asked about solitary confinement. It has many of the same negative effects, but I'm very cautious about saying that's parallel because those are not randomly selected individuals either. They're on solitary. Is it tied to recidivism? I would suspect it is, but I know of no study that's looked at it. But I would certainly suspect that it is. Yes, go ahead, and then you.
So how do you address loneliness, or what can we do to lower our levels of loneliness? That's
a long answer. It's actually a very, very important question. I thought we started this meta-analysis a year ago. It's called it's quantitative analysis of all the extant literature. We started this over a year ago because we started with just under 1,000 papers. So it took us a long time to actually get through all of this. We did the meta-analysis, 5 qualitative reviews. That is, they weren't quantitative, they just read a subset of the studies. They all said group interventions are good. And so we thought, let's do a quantitative analysis and find out for whom, using what procedures, at what lengths, and what kind of groups is best. What we found was nothing out there is effective, zero. Now, we have a very different theory about what loneliness is. The model that I just told you, remember, they used to think loneliness was like this depression or introversion. It isn't. It isn't. So we have a different approach. We're now testing it. We're starting to develop materials, and we've got it in a laboratory, in a clinical laboratory right now, and I will see the second patient who went through on Monday. So we hope to be doing that research, crank it up a lot this summer. We just finished the quantitative review. But we do have an answer. It hasn't been empirically tested, but it's theoretically based. It may work, but we need something. I'm sorry, it's you, then I'll come back to y'all.
At the University of Chicago, Junction has been involved in this study.
Yes. I'll tell you, we did a study, so in 1980, there was a study of American and Chinese loneliness. I replicated that study this year to look at American and Chinese levels of loneliness, and we used University of Chicago studies in the American, and we found stunningly high levels of loneliness here. And I thought, is that University of Chicago or is that America in 2009? So we replicated the study using an Ohio State sample, and it was much lower. I love our undergraduates. I truly do. I love our undergraduates. But they're not exactly representative. They're not randomly selected, as you well know. I think. Okay, and I'm happy to stay up here, or we can talk outside. But let me ask you.
One tidbit on an earlier slide: effective mortality, low versus high connectedness. There was one county where it separated out white versus black, and the line for black is basically flat, where there was a significant effect for white.
Right.
So is race causative effect? Is it genetically selected?
We find within African Americans, we find the same pattern as we find for Caucasian and Latino Americans. We actually find loneliness to be the more operative variable. That was actual physical contact. And again, objective isolation isn't as clean a mapping as subjective, perceived isolation. So that's the reason I think that existed. You can have a lot more integration and you may feel isolated. By the way, African Americans feel more isolated than Caucasians, and it's primarily the collective. That is, if you feel like you don't belong in America, it's like those bears. There's a sense in which you don't have the same connection to the country. Okay, I'm happy. Thank you very much for your time. I'll talk to anyone else.